Hello students, welcome to EPG Pathshala. I am Indira Ghosh from JNU. Today we are going to talk on module Origin of Force Fields under the topics paper called Computational Biology. Learning objectives are what these force fields come from and what are they in reality, that is, what are these things and how are they derived. That is, when we have the macromolecule to explain how we get the force fields. Now, I would like to explain now, and that is the question you would have in your mind now is these parameters of force fields actually how it looks like. In reality, if I want to find out some parameters, how I will find it. Now, how they are written. We would like to hear specifically objective is to find how we have these force fields, this concept of force field. What is the origin? Actually, the explicit force or field or potential surface or potential energies has to be calculated by quantum mechanics. As it has been described here and statement by Paul Dirac, 1929, that these physical laws necessary for the mathematical theory of a large part of physics and whole of chemistry are completely known, and difficulty is the only that how to exact application of these laws. So we know the laws, but what should be the equations by which we can calculate all these values? The calculation method is only can be done with the equations, and these equations are much too complicated to be solvable. Hence, big molecules like macromolecules, protein, DNA, carbohydrate, lipids, etc., or membranes, and even aggregated complex molecules become very difficult. Hence, he continues to state the practical methods of applying quantum mechanics has to be developed. And please note, he has mentioned that a practical method must be developed. And this can lead to an explanation of the main features of these complex atomic systems without too much of computation. This statement has turned out to be one of the most important statements to look at today's world, large macromolecular interactions. Basically, it states that we are all doing modeling, hence accuracy we might not get as much actually the system may behave. However, having an approximation between quantum mechanical things and classical mechanics, we might be able to solve this and find the interactions. Here, today I will go through with you these explanations slide by slide, how the quantum mechanics, without compromise, we try to go for the expression of force field and potential energy surfaces by following slides. Most well-known quantum mechanical approximation is called Born-Oppenheimer Approximation. What it allows you to do is, standard time-dependent Schrödinger equation is shown in the top, which is H psi equal to IH del V del T. However, this time-dependent Schrödinger equation can be divided into two parts. One is time-dependent, another is time-independent. As the system, what we deal with are a long-term equilibrated systems, we do not need to know about the time dependency. Hence, time-independent potential function is quite sufficient to explain for our systems what we are going to talk today. Born-Oppenheimer approximation actually separates this motion physically, that is, nucleus moved much more slower than electrons. Hence, the motion of electron can be actually neglected with corresponding to electrons' motions, being very small, much smaller than the nucleus. Hence, we can estimate all the chemical reactions or chemical interactions even by only depicting electron motions and removing these nuclear motions. Hence, the following expression becomes H psi d psi and Ed, which is a function of all the electrons only, with psi, which is also function of electrons. If we can express this E function of all the electrons, approximate it or nearest to the natural potential surface, we can actually calculate the total energy interaction between this system, which I will try to explain to you in their terms. However, one must remember in many reactions like nuclear interactions or nuclear and electrons interaction, etc., chemical reactions like exchange of electrons or from one shell to another, some of those will be limited application if we use this potential energy surface as only electron dependent surfaces. How we parameterize this electron potential surface? So, force field is one of this specification, which is a mathematical concept to explain these potential energy surfaces. Additional these properties of any atom, such as connectivity, hybridization, oxidization state, etc., requires the specification for force field. In addition, we should be having the atom types, that is, each atom has different kind of electron shells interacting with the covalent, ionic, or even non-bonded interactions. So this participation of atom types is important to parameterize the force field. Actually, what we require is to find out the force field. We should have general parameter set like atomic mass, van der Waals radius, partial charge of every individual atom, equilibrium values of this bond length, bond angle, which I have shown in previous module. And we should have the spin constant when we use the harmonic potentials to depict each of these deviation of structures. This is a function used in amber force field, which I am showing here. In this module, we will discuss more about how we get these functions. The very reputed form of force field is given by amber, which is called assisted model building and energy refinement. By the term itself, you must be very careful and understand that we have done something called assisted model building. This is not actual structure, but it is the model which we are building, and we are calculating these functions and force field for energy refinement. That's limited to only calculation of large molecules functions. If you want to calculate very thorough energy, you have to use quantum mechanical systems or quantum chemical systems. Here we use more or less rather empirical functions, which is forms are written here, functional forms we call them, and it has been displayed even earlier module quite a lot, what they mean, each of the term, how they would got this expression as well, and how nearest they can be depicted to their function actual. So here is the full form of amber restricted functions, where first on the line of the first top line 3 functions are depicted expressions for bonded interaction, whereas the bottom line is depicting the non-bonded interactions. Especially you can see in amber, hydrogen bonds are explicitly taken care of. Hence, amber has this facility of including the hydrogen bond functions in your model building. How we get the parameter sets, and that is the constant values like P0s, Ks, etc. These are collected for protein, peptide, and nucleic acid, etc. And there is a standard format for naming them, which begins with FF, particularly in amber force field, and contains 2 digits, that is contains its year number, and then the modification versions, that is how many new versions have come. However, amber force field also includes not only protein, nucleic acid, and carbohydrate papers, but it actually does not include any small molecule, which is separately created by GAFF functions. And this module is also available in amber force field to calculate for small molecule like ligands or chemical novel, ideally ligands which we are designing. Additional force to explain carbohydrates are also available for glycam force field call, and that is also very important. So using those force field, one can get proper carbohydrate geometries done. The torsion angle parameterization in the AMBER. Parameterization for torsion angles have been done under AMBER, and so all of them has been done under other softwares as well. I have taken one example which is more popularly used. That's why I'm using AMBER. And in AMBER, torsion angles are contains one term and from the cosine expression. But to accurately model these bonds, it has found necessary to include more than one term, and that's why sometimes one uses three terms for expression of proper torsion angle. Like the example is given here, correctly to model torsion between carbon-carbon containing two oxygen at the end, one needs to have potential function which is depicted in the bottom. And these are actually developed by the scientists working in actually they dedicate their time to develop this force field to actually explain the functions which experimentally already been known. And that's why the newer form of versions of these potential functions are coming out, or constants and others. In AMBER, another interesting point is use of general torsional potentials, which is like this examples, and energy is solely dependent on the two interior atom, not on the exterior. That is, it is independent of whether the carbon is bonded with the externally hydrogens or not. It only matters, most often it has been fitted in such a way, only matters the central bond through which the two planes are rotating. And this facility is available in AMBER and also in some other software as well. However, now I come to the most crucial points about these force fields and potential energy surface. We have developed mathematical theory basically, and this must have, and that's why from the look at the structures and the functions and parameters have been suitably chosen, that it must have multi-body potential, which I have depicted in my previous module, that is one body, two body, three body, etc. It must have a transferability, that is, it should not depend on protein separately or molecules like small molecules separately. This function should be, energy function should be transferable all across every type of chemistry. Variation in geometry due to environment must be taken care of, because this is where comes the biological molecular functional ability. Most of the interaction in biological molecules are really due to its non-bonded interactions and environment effect. Thermodynamic perspective must be there so that we can use the molecules and collect not only one conformer but ensemble of conformer. But the best part of these functions are that they all are additive. Hence, if it is necessary, we can actually take new functions and just add it and get our potential energy surfaces. Also, being self-consistent method, it has many new atom types. Only thing we have to do is we have to introduce that atom type, its bonded interactions, etc. So there is no limitation of what kind of models we are, what kind of chemistry structures or chemicals we are using. And it has been expanded so much so far that it has expanded to nanoparticles, new molecules, etc. The force fields are applied. This is the novelty of such a system which has been developed over last 40 years. Now I'll come to what are different kind of force fields available. There can be all atom force field, which will include every atom, including the hydrogens. But most often these force fields are not very useful for explaining the macromolecular structure. However, they will be very important if you are trying to explain vibration and spectra, etc. So most often people use this united atom force field. It is abstracted the hydrogen and carbon atoms as single interaction center and adjusted to particular mass, charge, everything, and even including all other stuff. However, you must remember While doing this, nobody has forgotten about hydrogen bonds. So NH or C double bond O or COOH kind of interactions which are, or sulfur hydrogen, all those are exclusively considered as the functions, as we would like to look at hydrogen bonded interactions. And lot of cases hydrogen bonded interactions plays important role in biological molecule, and they are not considered as united atoms. There is another kind of force field when you would like to see only the backbone or only the topology of a molecule, how it's changing. One can use something called coarse-grained force field, in which one would like to use not every atom, but maybe one set of atoms, maybe whole peptide bonds, maybe one peptide unit, etc. And we will not discuss much of this, but we will specify our united atom force field. That's what regularly one uses for assigning or calculating different kind of informations. Various types of force field. I would discuss here what are well-known names of different kind. One is the crudest model could be lattice model, where actual force fields are yes or no kind. That is, the molecule can stay in one conformer to another conformer. That is the most crudest type of force field one can use. And no chemistry is preserved much with it. However, next onwards, the other force fields are united atom force fields, where OPLS, one of the optimization force field, has been used. Molecular mechanism force field has been used. Amber and Bromax have been used. Then comes another elaborative position for using all atom force field. CHARMM, OPLS, AA, Amber 94 onwards, they use all atom force field. Of course, we are still developing a few more new types force field, which is to explicitly use polarizability, and that's called polarizable OPLS AA or Amber 2002 onwards. Tinker PFF. These kind of force fields are specifically used when you have electrostatic role to play of the protein is major. Hence, one must understand that usage of force field or choice and selection of force field depends on the case study you would like to understand in macromolecules. An obvious reason, the amount of computational time increases with this newer or more terms included in the force field expressions. This slide actually historically sums up the different kind of force fields, and mostly the relationship are shown in everywhere. Only a very small differences have been found from class 1, class 2 or class 3. However, it depends on how much we are looking at intricacy of functionality of protein. Most often, the class 1 does not include any coupling between the terms. That means no coupling is used between bond angle or bond length or bond angle and torsion angle, etc. And those are called class 1 potential force field, and earlier slides contain one of the samples. Class 2 actually includes anharmonic terms like Morse potential and also cross terms to include the coupling between internal coordinates. And this can describe a strange geometrical system very well. Hence, in many cases where we have carbon nanoparticle or constrained geometries, one may use this class 2 type potentials. However, in description of electrostatic, if you want to go in detail, and it plays more a major role in our functional understanding, we must use maybe class 3 potentials or functions, where you include hyperconjugation, electronegativity, and polarizability if necessary. So, as an example, this slide shows you from the CHARMM definition. The CHARMM is the Harvard package for doing simulation and energy calculation for macromolecules. The parameter like mass or for calculation of kinetic energy, you require mass. Hence, each of the atom types are shown like this, which is depicted here. The number which is written to 20 and C, that means the 20th atom, that's all. Now, that is a serial number. However, the mass is given as 12.01100, and the definition of carbon is C. That's the used in the term in the programs. Now this C refers to carbonyl C, also in the peptide backbone C, that is C alpha. Now question is that these kind of values and their definitions are very clearly written in every slide which I will follow through. And this shows you that if you want to introduce some other terms, you should be consistent with this formula or this type of parameter files. In every software, always you will have separate parameter file and topology file or residue topology file, which contains all the parameters defined already. Example here depicts as how is the residue definition in one of the software's package or potential function is in CHARMM. CHARMM has been developed by Harvard University and available for users, friendly way to use the software. However, this is one example, and most of all the softwares of packages which it uses almost the similar forms. Here I show you the residue of alanine. If you want to introduce a new residue, a new small interactive molecule or a ligand, you can make the function look like this. Here it actually tells you called residue topology file. It contains a part like this, and it shows you every detail, as I told you, all the atom, how their connectivity is, how much is their fractional charges, which is shown in the fourth column, and also it says which atoms are bonded to whom. This even it specifies which are double bonded or single. So this kind of information in a text forms are given in the residue topology file for all 20 amino acid, and even more than that, all the nucleic acids available, etc. So to build the molecule or the protein structure or nucleic acid structure or DNA, RNA is very easy from this internal information. So this is depicting in about the structures. Next slide will show you what about the parameters, which is related to atom types, which is defined here. So one must remember these numbers, NH1 or CT1. These are atom types are used in the next ones, and even sometimes even the second ones like this. Now, please do note, most often the residue topology files are very much goes hand in hand with protein data bank formats. And there are programs available in the software which can translate it one to another. Here I'll show you how the amber atom types are taken care. But please note that CT, which I showed you earlier, still the same CT is used even in amber. Here actually it tells you what are the atom types, declares all the atom types available in the system and what are their functions, how their chemical or their available electrons in their external orbitals. And that helps a lot. I'll show you a few slides like this in few next, which depicts considering each of these type, number, and their bond lengths, bond angles, etc., some other parameters. Earlier I showed you the amber force field, and here I show you the amber force field's bond parameters. It's listed exactly how I have taken out of the program. So program has a database, and in that it has been depicted. It's been included in it, and it's been in 3 columns as you see. First column gives you the character of those 2 atoms, their nomenclature followed as the atom type file follows. Second column follows all the values, that is the constant, the bond constants or Kb. And the third value is the value of the distance exactly in angstrom, that is B zeros, which has been parameterized in the expressions. All set of atoms are given in this file, and then you go to the next file which gives you angle parameters. Here you have seen here would have atoms which are given on the list of atoms, how they are connected is given on the left hand first row column, and the second column gives you the same thing, k theta or constant, and the third gives you the values as equilibrium structures of crystal of containing such molecules have given. And this is how repeated in all like torsion parameters, which also gives, in addition to those three, it also gives the value of n, that is how many fold. Like sp3 will have threefold rotations, hence the value of n is also here, and that's how it has been written. One more column comes here. However, please note, most of the nomenclature uses the bond through which the torsion is done is put it in the center bond. Now I show you here the improper torsions, which is using the same atoms, but very interesting is the bond through which it is rotating is shown. One picture here should be shown to be considered here. And if you notice, all of them are irrespective, and this is a function taken from Amber, and all left hand is xxx or even anything, because this type of improper torsion are dependent only on the connecting bond through which the rotation happens, which is shown here. Okay, so improper torsion values are shown here, and next slide shows also the another set of van der Waals potential and others. Van der Waals interactions are much simpler because it just takes care of two atom types, which is shown here, and the two parameters. One is their sigmas, that is the minimum size of site where is the lowest energy possible, that is the interactions can be both together and favorably, and the depth of the potential well. Those two functions are given here so that one can calculate the van der Waals energy Linac-Jones parameters from any of the systems. Next slide shows us how we consider, or in at least in Amber, the charge or partial charge. Most preferably, we use the partial charge, and that reason is the Coulombic potential should not be too much exceeding and highly, because we do not use higher order. That means we do not use dipole moments, we do not use multiple expansions. So it's better to have a low charge and ASP fitting. And Amber uses potential energy fitting, that electrostatic potential energy fitting terms of each of these amino acids and from the quantum mechanical calculations, and produces a very good partial charge list, which is shown here for glycine, alanine, leucine, and isoleucine. We explicitly wanted to address the hydrogen bond function. Hydrogen bond is one of the most important molecule bond, which is very much useful for functional purpose of proteins. And most of the protein structures are even stable due to this internal or external hydrogen bond. Like in alpha helix, it is the helical hydrogen bonds which stabilize the structure, whereas in beta structures or pleated sheets, between two different distinct external hydrogen bonds makes the stability of protein. So in nucleic acid, the bases are bonded by hydrogen bond. So ample example shows that the importance of hydrogen bond must be given in the energy functions. What is hydrogen bond? Hydrogen bond actually are between two ionic or polarized interactions, and being polarized to two adjacent, mostly it has been found with water molecules in the initially. One can see the geometry how it looks. So oxygen with the water molecules and the other water molecules can be available for binding, and electronegativity of oxygen attracts the electronegative. So one of the oxygen of water becomes more electronegative than the others, and it shares the hydrogen for a short while. And this is interesting because it's a hydrogen bonds have hardly 1/20th strength of a covalent bond. However, hydrogen bonds do have one very interesting property called cooperative property. That is, many hydrogen bonds are always added the system to become more stable. Hence, it is one of those very important hydrogen bond formations. And the best hydrogen bond, or the strongest hydrogen bond, only forms when the three atoms, as shown here, who are participating. One is donor, another is acceptor, and the hydrogen is on one line. That means all the angle between OHO must be 180. Here I show you the different kind of water hydrogen bond possible, and it can give you that even at 37 degrees centigrade, 15% water molecules are joined with 4 other short-lived hydrogen bond called flickering cluster. And people have really researched on, especially when you go to the ice formation, these hydrogen bonds plays a strong, stable structural behavior. Please note, the properties like high surface tension, specific heat, heat of vaporization being very high, are all the properties which comes from the formation of such hydrogen bonds. So, as it shows to us that hydrogen bonds plays very important role, we would like to look at what should be the parameters of the hydrogen bonds. Next slide shows us that. Hydrogen bonds simply calculated in most of the systems as partial atomic charges where oxygen, which is the acceptor, becomes a little more electronegative by 0.5 part, and the carbon connected to it or any main chain or side chain atom connected to it becomes a little more partial atomic charges positive, that is 0.5. Also, the donor atoms has hydrogen carries a partial atomic charge of 0.35, and the nitrogen or oxygen or any carbon or even carbon sometimes becomes electronegative, more electronegative than normally it is. And this is only changes happens for the extra other function which is added in the functions of energy expression or force field expression. This is very good to understand that how one really calculates when you are computerizing the whole program system, which is shown in the next slide. We would like to look at, this is called interaction terms or alternate interactions terms, where explicit hydrogen bond is shown. Amber calculates hydrogen bond in this manner. Here, not only the term of between two donor and acceptors are given in a 10-12 potential function, in addition multiplied by the cos theta between. So the strength is in the aligning 3 donor, acceptor, hydrogen and acceptor line. Hence, the angle is also explicitly taken care. Of course, the hydrogen bonds, when it is taken care of, that we should not calculate their van der Waals radiuses, etc. So, in those terms which include such function, van der Waals radius or Linac-Jones potential for those pairs are used in a different way. One is 9-6 potential, sometimes the other way around. These functions are used selectively in where, like in amber, but explicitly used when one says, I would like to use the potential energy functions explicitly for hydrogen bond. Now, as I have shown you the structural parameters earlier, along with the KBs or K thetas, which are actually experimental. Now, the question is, where this comes from and why we need it, because if we know where to do the new parameters, we can generate when we require to use in our real proteins, DNA, carbohydrate in those cases. So, I am going to now speak a few slides and show you how these parameters are derived from. I'll come to the point now, final things like how we parameterize these force fields. That is, if I do not have any unknown, how I am going to get it. Few slides now will explain to you that functions. Equilibrium, bond distance, etc. As I have told earlier, X-ray crystallography will provide you. Bond and bond angles, force constants from vibrational, infrared, other spectra, or calculate the normal mode with quantum mechanics. Dihedral parameters also are based on experimentally or fit from your quantum mechanical calculation. Atom charges are mostly calculated by using quantum mechanical high electrostatic potential, high level quantum mechanical calculation, and fitted by ESP or electrostatic potential function. Linac-Jones potentials are also determined from experiments, mostly from liquid properties. Next slide shows you the actual results of or the actual parameter. This force field, if I want to find a new force field parameter, where I should look for, and this is also in a very good book to study, is given a reference, and Internal parameters like bonded parameters, you have equilibrium constant, multiplicity, phase, etc. The properties are found because of the vibrational and conformation properties and sources. External ones, we can have pure solvent, crystal structures, etc., interaction energies, Brien-Rosse interactions, and other systems, which all are given here in a total number. And it's better to understand our functions because they behave better also, and when we correlate with experimental properties, they go hand in hand. The most important reason is that we actually use parameters which are coming from the experiments itself. Here I just want to address this question which every time students always like to know. So there are several types of popular force field, and all of them are also available most often for academic institution without any very meager small amount. Which force field I should use? It all matters how rigorously you want to calculate. However, one must understand, if you are calculating any system with one of the force field, you should continue to compare all your data with the same force field. Otherwise, there is a very much difference because every force field has a set of different residue topology file and different parameter files. Here I have just given you a list of many of those like CHARMM 22, recent number is 34. AMBER 94 started in the beginning, now it's 10.0 AMBER. And actually it depicts how many different forms it has grown up since last 25-30 years. Now GROMACS is another one which has been developed in early 90s from GROMOS 96, which used to be in Fortran, and then it has been converted into C languages. The most recent GROMACS has become 3.3.2, but maybe by the time you listen, these numbers all changes. All of them has a historical perspective, like scientists who were working in the field, like CHARMM at Harvard were developed. Martin Karplus is major force behind, and his large team of people who work even today. Alexander Mackerel is looking after the softwares and many of others, and developing such a huge amount of parameter and receptor files. AMBER was developed by Peter Coleman and now looked after also by a large group in Scripps, and it's been very effective and useful for drug designing, molecular simulation, etc. with the protein, nucleic acid, and carbohydrate. GROMACS, on the other hand, was developed mostly at Bredenson's lab and later on with Van Goolstrand's lab, and converted into GROMACS, and many users and effectively many developers are looking after this force field. So one can use any of these with what the purpose. A series of them are also available in OPLS-AA and AMBER OPLS together. Sometimes two different force fields are also combined together and provides us these facilities. And there are certain other specific work to be done, like polarizability or solvation energies. One uses such force fields. I have now, one of the slides I have shown earlier, that the force fields etc. should be actually showing the effect of environment. Here is a beautiful picture of a molecule, that is a ion channel, which is embedded in protein and also many of water molecules. So in reality, one would like to take the picture of these values, that is how the protein, if it is inside the membrane, how its conformation will be affected, or if it is having a solvation, that is it's dissolved in water. In most of the cases, cellular dissolved proteins are surrounded by water, how the parameters should be included. Now, I would go through few slides to explain to you what are those parameters and how one handles this kind of simulations. Here I am just showing you one example how the solvation free energy can be calculated and compared with the values experimentally known. Paper reference is given. So Every amino acid one can do simulation and finds the solvation energies and plot them exactly like this. And one can actually take three different, here it's three different values have been taken, the three different force fields. And if you notice, and there is a one which is experimental values. If you notice this three different force field, you can see there are discrepancies in the force fields and output. Some of them, as you see here, some of them fits very well experimental values, but some of them are a little bit off away from experimental value. However, if you notice carefully, you would see most of the experimental values tallies for those like ALA, VAL, LU, isoleucine, etc., which are hydrophobic groups, or solvation is very less. But this is the proof that every function or force fields are unique to itself. Hence, combination of these force fields are never suggested, and it may lead you wrong ideas and wrong concepts. So please remember very strongly to continue to use only one type of force fields and compare between them. That's it. Many papers has come out to justify how many force field and which type of force field are very appropriate for DNA, RNA, carbohydrates or proteins. I would like you to go for a lot of references, and ultimately one can look into them. How we use the force field, that is the most important to biotechnologists and biologists. Actually, what we can do is we use the energetics of biomolecular structures, that is the interaction between them and stability of them, etc., and also binding ligands between each of these preferable protein structures. It is also applied very extensively, mostly for molecular mechanics, for understanding this. Also, most importantly, solvent effect, which is the very relevant, and we will address them at the very end. However, not only the energy like enthalpy is important, in some cases even entropy is very important. So how to find enthalpy separately and entropy can be also demonstrated by molecular dynamics at the end. These are three major important things to look after when we do biomolecular structures optimization, interaction, and functional detail. Here I depict how many applications. The major application is done by X-ray and NMR structure refinement, which is one of the greatest help for most of others. We have also protein structure prediction like the stability, etc., folding kinetics and mechanics, dynamics of conformations and how it affects the functional problem with the or functional behavior of a protein, DNA, their interaction between, optimization of their structures, simulation with DNA and RNA structures. Also, we are able to do even simulation with large membrane protein, which are not so easy to get crystal structures. Molecular simulations can also show you NMR structure refinement as I have described. Here is some show for protein structure prediction with two protein interactions, protein folding, pathways, different structures, global optimizations, also the effect of conformational dynamics. These can be learned really if we do the molecular simulation, which I would discuss in next modules. So, let us summarize what we have learned in this module. Actually, we understood the definition and origin of force field, which is explained, how the parameterization of this force field can be done, also the different type of force fields. Not only that, we have taken an example from actual database of the force fields, parameters and residue topology files from AMBER and CHARMM, which is most popularly used, are shown here, and how the format of the files also are shown here. In addition, this is a concept which has been applied to molecular simulation and molecular dynamics, and few examples how we can actually understand that has been shown pictorially here, including the solvation effects. And next module would address how we actually perform in reality, the simulation of large molecules using these force fields.